right, I'd like to introduce our next speaker, Mike Boland from UPenn. He is also an STXBP1 dad and a member of our scientific advisory board. Mike.
Thanks, Charlene. It's a pleasure to be here to speak with members of my fellow community. I often speak to academic and biopharma audiences, and I'd always like to include this collage of STXBP1 kids just to remind them that there are faces behind the work that they're doing, the treatments they're developing, and biomarker research and whatnot. And it's not just because my son Lucas is on this collage, but I think it really does help drive home that there are real people that behind a lot of this work. I am the strategic director for translational and clinical research. I was at Columbia University for a number of years, developing gene-targeted therapies for STXBP1 and also studying other pediatric epilepsies before being recruited to Penn and CHOP. So Jim gave us a great introduction to biomarkers, and I don't want to belabor the point, but I want to emphasize that a biomarker is something that we can measure that can either predict disease status, progression of a disorder, or a response to treatment. And they have different names: diagnostic, prognostic, pharmacodynamic. And I'll walk you through a little bit of our efforts to identify treatment responsive pharmacodynamic biomarkers for STXBP1. And there are two flavors to these biomarkers. Molecular biomarkers are things that we can measure from inside tissues like blood or cerebrospinal fluid, not necessarily tissues, it's biofluids, biomaterials. And then physiological biomarkers such as qEEGs and biosensors. And for any of you that have been involved in the STAR study or potentially ESCO, I'm going to show you a little bit about how we utilize all of these data to identify different molecular and physiological biomarkers for STXBP1. And I had to also include cute photos of Lucas getting qEEG, as well as using the biosensors that we use to measure tremor, which is something that Ingo Helbig alluded to earlier. So we're going to talk a lot about protein biomarkers. Well, what is proteomics? You hear about proteomic biomarkers. Proteomics is the comprehensive study of the proteome, which is the collection of all of the proteins that are expressed in a given tissue or cell type at any given time. There's a couple of different types of proteomics. Structural proteomics can give us an idea of the 3D structure of proteins, and actually there was a Nobel Prize awarded a couple of years ago to AlphaFold for its AI algorithms to pretty accurately predict the 3D structure of proteins, which has been a really big drawback to therapeutic development in a number of different disorders. And so I wanted to show this is the actual protein structure of STXBP1 and its binding partner, syntaxin. And then once you have these protein networks, you can really start to understand how how your target protein interacts with other proteins, and you can start to think about other proteins that could also serve as biomarkers if you can't accurately measure your target protein. And so expression proteomics is what I'm going to talk about most today. And this measures the abundance of your protein of interest in a given cell or tissue or biofluid. And then they come in unbiased, where we profile all of the proteins that are expressed in that tissue or biosample, or targeted. This is more like an assay that would be developed specifically for STXBP1 to study it at study its levels at very high resolution, almost like a diagnostic assay in the clinic. And together these will give us an idea, hopefully, of a diagnostic biomarker. We can see haploinsufficiency in blood or CSF, and also pharmacodynamic, where if we can detect haploinsufficiency in blood or in CSF, then if we give a therapy like an AAV-mediated gene therapy, hopefully we can then use that to see if we can normalize the STXBP1 levels. And so let's start in human CSF. And we first turn to, can we detect STXBP1 and other relevant proteins like its binding partner syntaxin? And to do this, we turn to an unbiased proteomics approach because we spent several years trying to develop a very targeted assay to detect STXBP1 in CSF. And it was incredibly difficult, and we had just like failure after failure after failure, and then decided to use this. It's called the SEER platform. It's a nanoparticle enrichment where each of these different nanoparticles has a different physiological property, and so you incubate these nanoparticles. It's a milieu of over 100 or 250 of these nanoparticles, and they all have different binding affinities for different proteins. And so the proteins in your plasma or your CSF bind to the nanoparticles, and then you can extract the nanoparticles and study the proteins in more detail. And I just show you an example of some of the 6 different samples of normal pediatric cerebrospinal fluid that we studied from the CHOP biobank, showing that we can accurately identify thousands of proteins in a relatively consistent manner. And so obviously, we're interested in STXBP1 and syntaxin, and we were shocked and very encouraged to see that not only can we identify STXBP1 and the 2 different isoforms of syntaxin 1A and 1B, but we can identify them at quantifiable levels, even in CSF. And this is at very low levels of CSF. We're also interested in some of these other secreted neuropeptides, because when we think about STXBP1, we almost always talk about STXBP1's role in regulating synaptic vesicle exocytosis and its function on neurotransmission, its communication between neurons. But there's also another role of STXBP1, and that's in this dense core vesicle release, and that's through binding a different syntaxin. And these neuromodulators you see as these little black balls. These are secreted proteins in the central nervous system that are signaling molecules that actually help maintain normal neuronal functioning. And so, because STXBP1 releases them, we would expect to hopefully potentially use them as a secondary biomarker if STXBP1 isn't necessarily available. And we're encouraged again to see that a number of these different secreted neuropeptides are also detected at quantifiable levels in CSF. This experiment was just to demonstrate that we could detect a consistent STXBP1 and secreted neuropeptides over time from samples taken from the same patient. So we went to the CHOP biobank, which contains a large number of samples of kids with SMA who are coming to CHOP to receive their ASO through spinal tap, through lumbar puncture. And every time they do that, which is every 4 months, they take a little CSF. And so we have a really large bank of CSF that we can draw from. So we took three kids with SMA and we profiled their CSF at 1 month, 4 month, and 8 months. So that this was extracted from them at say 0 month, and then 4 months later, and then 8 months later. And what we can see here is that we can very reliably detect STXBP1, the syntaxins, and these neuromodulators consistently over time from patient to patient, which gives us a lot of confidence in this approach. One of the most useful things about this unbiased approach was it told us when you're doing proteomics, you're not measuring the whole protein, you're measuring little parts of the protein. And some parts are more measurable than others. And using this unbiased approach, we were able to determine which parts were the most measurable for a particular method called mass spectrometry. And that has enabled us to start to develop a very specific diagnostic assay for detecting STXBP1 in the CSF. And it's similar to what I'll talk about here in plasma, because it's the same peptide. 5 minutes? Have I been up here for 15 minutes? All right. Why blood markers why blood biomarkers? It seems counterintuitive because this is a CNS disorder. But when you think about all of the crosstalk between the cerebrospinal fluid, the lymphatic system, and the blood circulation, there's actually quite a lot of crosstalk there. And so we can use blood as potentially a peripheral signature of a central nervous system disease process, and try to study proteins that are essentially excreted into the cerebrospinal fluid that then make it up into the blood. This is a study that Jim alluded to from Combined Brain, and we've further refined our analysis and detection techniques to identify STXBP1 in the blood. And I won't go into the nitty-gritty details, but I will suffice it to say that when we look at STXBP1 with a very specific, almost diagnostic assay, we can very accurately determine haploinsufficiency in blood from STXBP1 patients compared to controls. And this is definitely, as Jim alluded to, a numbers game. And so in the beginning, we weren't able to detect haploinsufficiency because we weren't powered enough. We didn't have enough control blood samples. As we increase that number of controls, now we can start to very accurately determine haploinsufficiency in the blood. So I will spend very quickly amount of time. You just kept going, so I'll just go. I want to talk, this is more about what you guys see more often, what you do in the STAR study. You come in, you get your QEEG, and recently in the last year and a half, your kids have been strapped to biosensors, and we get a lot of data from that. I won't go into this in detail, but suffice it to say that clinical EEG that almost all of your kids have had is a little different than research QEEG, which is just a cap that goes on and we do like a 20-minute recording, as opposed to electrodes that are glued on the head and you do either a 24-hour or a 1-week reporting if you're in the epilepsy monitoring unit. And using these QEEG tools, we've started to develop an analysis pipeline where we can begin to tease out disease-specific QEEG signatures that we might be able to use as outcome measures or biomarkers or even endpoints in future interventional trials. And here's an example of this here, where our team at CHOP, led by Jill McKee, has identified a number of different QEEG features specific to STXBP1 patients. These are not seen in other, like 4 or 5 other disorders that we have looked at: SYNGAP, Dravet, SCN2A. And what it is the frontal alpha theta power is reduced in our kids with STXBP1, and this is consistent from toddlers to children to adults. And the light purple are the clinical STXBP1 EEG scores, and then in the dark purple are our research QEEG scores. And what you can see is they really largely match each other, especially in children and adulthood. So what we think here is we have a really like a specific spectral power phenotype for STXBP1 that's really non-invasive to measure. You can do it in 20 minutes. And based on all of the developmental scores and whatnot that you've heard a little bit about, the Bailey and all of that, we really wanted to see if we could correlate some of these EEG or brainwave spectral features to different aspects of development that we see in our kids. And actually we see that this frontal alpha theta power is very highly correlated with Bailey gross motor, as well as some other measures like fine motor. And so we're really digging into this and refining this approach. And then the last thing I want to talk about is these wearable sensors that I think is probably one of the coolest things we're doing, where we have a kind of a standardized assessment where the kids wear wearable sensors on their wrists, their upper arms, and now their trunk and their head, and as well as their legs. And we can record children doing a standardized reaching task to measure the very distinct STXBP1 tremor, which is a submyoclonic myoclonus, subcortical myoclonus, and then we can also measure. We're going to start getting into gait analysis and things. But I'll play this video, and this is Lucy Prosser with Julie Orlando, our one of our PTs, who is really spearheading this effort. And if it plays, you see Lucy reach for that object, and her reach isn't straight, and it isn't fast. It kind of meanders. And so this is what we want to measure in STXBP1. And this is my son Lucas wearing that. This is what we see when on the screen based on where the sensors are. And so you can see him leaning down to play with something on the floor, and then he sits back up. And so we can very accurately, using these movement metrics, measure tremor and other different metrics. And so here, what I'm showing you is an example of, on the left, typical development in this structured reaching task, where you can see we're combining a number of different. We look for at least 3 to 5 reaches, and then we can collapse the data and analyze it. And so one thing I want you to point out is at the bottom on the horizontal axis, you have that blue diamond, or is it? It's black there, it's blue up here. You can see in typical development, it takes 1.5 seconds to grab that thing. For STXer, it takes about 4 seconds, decidedly longer. Also, the acceleration of the reach is in typical development, it's up to 15. In STXBP1, we're looking at 6. So half the time, or half the speed. And then you also see how much variability and how much motor dysfunction is in the reaching task with STXBP1 by the number of squiggles in STXBP1 compared to the typical development. And then on the bottom is this is just quantified with statistics. And we see that for all of these metrics, except for the acceleration time area, but for all of these metrics, we see a distinct difference, quantifiable difference between STXBP1 and typically developing kids. And again, just like we wanted to correlate the QEEG profiles with developmental milestones or developmental metrics, we wanted to see if some of these different aspects of biosensor measurement that we can measure, such as on the x-axis, reaching duration, peak acceleration, peak count that we just talked about in the last slide, are these also correlated, or can they correlate with different aspects of STXBP1 development? And here we have P-body eye-hand coordination, P-body hand manipulation, the gross motor functional measure that Sam has talked about before, as well as Bailey gross motor and fine motor. And you can see that the reaching duration is very highly correlated with all of these gross motor metrics, which gives us a lot of confidence that what we're seeing could very well be a quantifiable, usable endpoint in a future clinical trial. So with that, I will end and just tell you that when you come to the STAR study and you donate blood, and you do the EEG and you do all of this, it is being used and put to very good use. And this is a very important part of the STAR study as it goes forward, because we're really getting into the point now where we've identified a number of these processes that can serve as molecular biomarkers or physiological biomarkers. But now that we've discovered it, we need to validate it. And it's in this validation phase that's really going to tell us whether or not it's going to be good for a clinical trial and whether or not the FDA will accept it. So please keep coming to your STAR appointments, and thank you for your time. We cannot do this work without the families and the patients. It just simply doesn't happen without you guys. So thank you for all of your participation. And I had to throw in Lucas. This was at the STAR study. This was our 6th visit. And thank you again.
Thank you, Mike. We have time for one quick question. Actually, come here.
So for an out-of-towner, and if I wanted to participate in this STAR research, and my son is 15, but right now he's in Canada and I'm here, and to get him here, it's almost an impossible task.
Yeah.
So can something be done long distance where blood can be drawn and from mom and me and him and sent to somewhere to get analyzed?
That's something that we're really working on now. And all of the blood draws we're doing are in clinic, but we are working on trying to establish protocols where we can do that and accept blood. We're also trying to work on establishing remote assessments.
Okay, go ahead.
So being able to do some of these standardized developmental scoring at home rather than having to come to the clinic, because
Okay.
we do realize that it's financially difficult and just difficult to travel with STXers.
Great, thank you. And my wife wanted me to ask you one question. Stem cell therapy, any value in that?
No.
Okay, thank you.
Yep.
And just to follow on, you can do remote blood collection through our relationship with Combined Brain. And
Yes.
so if you want to do that, come see Jim Goss or myself.
So stem cell therapy, no real benefit from that, and I'm a stem cell biologist. So.
Thank you, Mike.